happy to be here, and I will also tell you more or less the same as Manuel has told you now about my life, and then I will go on for more the research questions that we are looking at. Actually, I decided to study insects by exclusion principle. I didn't want to. So I will talk about it. I've also been working a bit on vertebrates, but then I decided that with insects you can really gain a lot of sample sizes very easily. So it is actually a very easy system to work with in a laboratory setting where you can really achieve getting statistical power because you get a high sample size in your research. So since say 2005-06 or so, I'm working on this topic that you see here. This is collective disease defenses in ant societies, and as Manuel has pointed out, we are in Maria Gurking, but officially it's called Klosterneuburg. That is a bit larger than Maria Gurking, which also has this Lourdes grotte. So. And this is the building that we were actually the first ones to move in 2010, because it's a brand new place for doing both theory and empirical work. The current focus is mostly computer science and biology, but we are also continuing and expanding in physics and chemistry. So it was an old hospital that has now received this first lab building, and currently we have 2 lab buildings, and in 2 years we have 3 lab buildings and so on. So I'm an assistant professor there. This means that this is a tenure track position. That means that I can be there for 5 years, and then I get an external evaluation, and if that is positive, then I'm getting a full professorship there. And this is actually a system that is not very common in Europe. So often, like for example, where I've been before at the University of Regensburg, when you have these university positions, or also at Max Planck institutes, you typically know that you have to leave after a given time. So this is then not a tenure track system, but you got a group leader position for like 5 years, 7 years or so, and then you know that you have to find something else. So this is a bit of a special case, and that made it also quite attractive for us to move here. And so I will just briefly always indicate what was my main focus. And here the group that I'm leading there is also called Social Immunity Group. And we now have 10 people in the group, 4 postdocs, 1 from Denmark, 1 from Lebanon, a German and somebody from the UK, and 3 PhD students and 2 technicians. Of course, if you have such a group, you also have to get external funding in, and we mostly got our money from the European Research Council. So the idea is that all the countries from Europe pay their money to this European Research Council, and then everybody from Europe can apply for grants there. And I got 1 big 5-year grant. I also got some Vienna or Viennese funding, which is called WWTF, where I'm collaborating with a group in Vienna. And because I had moved from Germany, I could also carry along some German grants and transfer them to Austria. But now they have been running out, but still part of the projects have been funded by the German Research Foundation. So maybe because this is a women academy, I should also point out that I also have 2 small kids, and that is also sometimes challenging, most of the time a lot of fun, but it requires a lot of organization to get it all a bit balanced and also get it all working, because my husband, as Manuel has said, is also at the same institute. But actually this IST is quite supportive. They have a kindergarten on campus so now the whole family is on campus, and they also learn some English there, and they get to know to an international setting, which I find actually quite nice for them. So previously I have been doing this German thing, which is called habilitation. and that means that this is something that means that one has been doing science plus teaching, and once you have basically the stamp that you have this habilitation, you are eligible for a professorship because you can prove that you have had a lot of teaching experience. And in recent years or so, this habilitation is not required anymore, but it's because it's basically what you're doing anyway, that you're doing teaching and science, you can also still get the stamp for it, so to speak. So it's actually not bad to have, but it also wouldn't have been a requirement for the current position. So this I did in 2006 to 2010 after my postdoc, and there I really got interested into this link of ants and disease. And I also study some particular ants, that is invasive ant species, and I will come back to that, why I find this interesting. More or less, when I started making this transition from my postdoc to my position, to this assistant position in Regensburg, I took a 3 months sabbatical, so to speak, because I was invited to join the Wissenschaftskolleg in Berlin, that is the Institute of Advanced Studies, which are inviting researchers from around the world to take 1 year off sabbatical, and there is no lab space or nothing provided, but there's only the opportunity to think about things, and the only requirements that you have is that you show up for lunch. That is easy, and for the seminars there. And it's very interdisciplinary, and it's quite interesting to get a bit a view of different disciplines, really covering everything. So there I started writing a review article, or wrote a review article, which means that one is browsing through the literature and looks what is currently known on a specific topic, and in this case we called it social immunity. So that means the social immune systems that we find in groups, that was actually for me extremely helpful, because you really get to know what the state of the art is in this particular topic, and you also take advantage of this and put it all together and try to develop what are the new things that you think should come out in the next years, what are the main research questions that are still open and should be addressed. And I was really lucky because I was part of this focus group on evolutionary immunology with really world-leading people in the field, and I really learned a lot there, and it was fun to be there. And before that, as Manuel has said, I was a postdoc in Copenhagen, and there I got interested in invasive ants. And for example, here is the invasive garden ant that is currently spreading through Europe. It was the first ant that has become a pest species in the, say, not so hot areas. So normally all these invasive ants were really restricted to, say, southern Spain or Spain, southern France, and also in the US to the warm areas. And here we now also have temperate climate zones that are suffering from these pest ants, and we were really covering the emergence of these invasive ants and studied their social structure. So this is interesting, I think, because if you look at native ant societies, you have a single queen that is typically digging a hole into the ground, and then the colony is growing, and the colony is of course never moving, right? So ants are very territorial, and they also have an area around the nest where they are foraging and that they defend rigorously against other species, but mainly against other colonies from the same species. So they often have single nest colonies, sometimes maybe 2 nests. One colony is so big that they need 2 nest space. But in all these invasive ants, they actually have merged to so-called super colonies. So all the species of a all the nests of this particular species in one population collaborate to one huge super colony, and the biggest described super colony is that of the invasive Argentine ant, which is covering 6,000 kilometers of non-aggressive ants across from Portugal, Spain to the French along the coast. So they would not be aggressive towards each other. And that's probably because they're all emerging from the same founding event. So I was interested to study the success of this invasive ant and also this particular social structure. And by dealing with these questions, the question also came up whether parasites have contributed to the success, because what happens is when you leave your native range and are introduced to a new place, then you leave all your diseases behind, because only the healthy individuals will survive the travel, and also there are very few individuals that are that start a new population. So this is often why they have an advantage in the new site before the local pathogens and parasites are actually adapting to this new host. And so before that I have done my PhD, and here I've already started working on ants, but there I wasn't not yet interested so much into parasite association with ants, but more at conflicts and fights in ant societies. Because what one has previously always assumed is that the ants are working nicely together for the benefit of the colony, and they are all for the society. And they also do that, they cooperate a lot, but what is a bit hidden under the surface is that there's actually a lot of conflicts going on also in these insect societies that often have to do with genetics. So if I'm more related to somebody or to my own offspring than to the queen's offspring, I try to maybe hide in a few own eggs and things like that. And so I have really looked at this in a particular case where aggression actually becomes quite overt, and this is in these male fighting Cardiocondylus species, where you can see here that an older male that is darker is patrolling through the colony, and whenever a new young male is emerging, and this is this quite pale individual because it's not yet sclerotized, it will be grabbed with these saber-shaped mandibles, and then they want to kill this rival male, but they don't make their own hands dirty, they just smear something on them, and then the workers are doing the dirty job of killing them. So they make really some chemical signal that they put on them. And I studied quite a bit of different questions with this system in my PhD. And I made this transition to ant societies because in my master's thesis I worked on grasshoppers, and in the end I decided that they don't really have that much interesting social structure, so I wanted to get more closer to societies, and that's why I swapped to the ants. Because here during my master's I was working more on hybridization in grasshoppers, so we had one major species and one species that occurs in the Austrian mountains, and how they are avoiding that, or like the small species, how it's avoiding that it's basically taken over by the very common species. And before that I did my biology studies in Erlangen, and I just wanted to pick out two points where I, in addition to the normal studies, also did some 2 or 3 months practicals. For 2 months I was a field assistant in Costa Rica, which was very exciting. So we were studying nectar-feeding bats. So quite a lot of flowers are actually pollinated by bats, like for example banana plants, and it was quite exciting to study them there directly in the field, but that is also where I realized that it's extremely difficult to get a lot of data because you have to be very lucky, and vertebrates, a lot of species are also protected. So if you want to find, I mean, everybody has to find the niche that one is most interested in, but one can also try and see what is more or less practical for the type of research that one is interested in. And so I knew that I wanted to do more lab work, and that's why I then swapped to the insects. And I also had another experience at the University of Kansas where I did my first real individual project on sexual selection in wax moth, and maybe that's where everything has started. That is then led to the PhD, because actually these males here are also fighting with each other. Not so interesting or not so obvious as the ants, but they are really trying to bump into each other when one male is calling for the female, so they are interrupting the calling or this wing fanning activity of males with which they are attracting females. So after this idea, so that you get an idea how one can develop a career, I would now like to really get to the science part. So I'm studying collective disease defenses in ants. So that means how is disease spreading in colonies of insects, and particularly ants, and what can they do to prevent these epidemics. And I'm interested in this link between disease and society, because if you're comparing solitarily living individuals with others that live in a group, and here I try to depict that this is actually the same for all individuals living in societies, not only ants, but also some parallels are also existing in human societies, then one lives in quite high densities, often performs frequent social interactions, often with closely related individuals. So that all leads to an increased risk of disease transmission, which can be seen as the cost of group living. But on the other hand, the individual defenses can be complemented by collective defenses, which can then be seen as the benefits of group living. So just to give you an example of societies, societies exist, of course, in vertebrates and in invertebrates, like in surrogates, in naked mole rats, and of course humans. And I'm particularly interested in the social insects, and there we have 4 different types. So on one hand we have the wasps, the bees, and the ants, which are quite closely related and are called the social Hymenoptera. Then we also have a second parallel evolution of social insects, which are completely unrelated, which are the social cockroaches or the termites. So their most close relative is a cockroach. I also would like to point out that I would like to differentiate insect societies from other groups that one could term asocial groups, because sometimes you have also temporary crowding of otherwise solitary individuals, like in migratory locusts or in processing caterpillars, and they are facing the same problem of high risk of disease transmission during this period of crowding. But what they are doing is not a collective defense, but an upregulation of the individual immunity, which has also been termed density-dependent prophylaxis. And this is very different from these collective defenses of social groups. So I would now like to tell you a bit more what I mean with these collective defenses and the levels of immunity that we can then find in societies. So First of all, each individual has its own physiological immune system. We are all aware of that. Vertebrates have an innate plus an acquired immune system, and the invertebrates have only the innate immune system. But nevertheless, they're actually capable of quite a bit of sophisticated immune defenses, like recently it has been found that they also have some kind of immune memory. But then in addition, each individual also has their behavioral defenses, like parasite avoidance behaviors and hygiene. And this can be summarized, the physiology and the behavior, as their individual immunity. When you then have several of these individuals interacting with each other, you can have this additional level of social immunity arising. And this is then the collective defenses of all the group members that are either performed as a joint effort or towards each other. So these can be either anti-parasite behaviors, or it can be the use of antimicrobial compounds, and it can also be the modulation of interaction frequencies. Because, as you know, diseases are spreading via interaction, and then one can actively modulate these interaction types and frequencies to prevent disease spread. So, similar to how an individual organism is fighting or preventing disease, we have a multi-step defense of this so-called superorganism of a colony against parasites. So first of all, the colony should avoid infection, so the parasite should be prevented from coming in. Then the parasite should be prevented from establishing, from multiplying in the host, and from transmitting to the next colony. So I would now like to give you a couple of examples how they are trying to prevent these different steps of infection. So for example, the leafcutter ants, they are very good in avoiding the intake of pathogens, because what they do is they have morphologically different casts. And you can see here that there is a really small ant that is traveling on the leaves that the large workers are collecting and bringing back to the colony to rear their fungus on it. And these small individuals, one thinks that their only task is to clean these leaves and to also prevent phorid flies from attacking them and putting their eggs on it, so that they don't bring anything pathogenic into the colony. Then wood ants, for example, and also the honeybees, they are actually actively going out to collect antimicrobial compounds like the tree resin and bring it into their colony and place it at different sites. And this is then reducing the risk of bacterial and fungal growth, and thus also the infection of the individuals in there. I'd like to come back to the leafcutter ants, which also have a really sophisticated waste management system, because to give you an overview of their lifestyle, they have huge nests underground, and more or less in the central areas they bring in these leaves that they are cutting, and they use this as a substrate to grow a fungus garden, and they are exclusively feeding on that fungus garden. And but that also means that they always have to bring in fresh leaves, and once the old ones are used up, they have to get rid of them. So they produce a lot of waste. And they thus have developed a lot of garbage dumps, and they can be also underground in some species, and in others they bring them out of the nest. And in addition, they don't only spatially separate the clean fungus garden from the dirty garbage dumps, but they also have a behavioral separation. Namely, they have particular fungus garden workers and they have particular garbage workers, and they don't interact directly with each other. So to avoid transfer of pathogens from the dirty to the clean nest part, these individuals are actually dumping the waste in a place where it's then later picked up by the garbage workers. And if a garbage worker tries to come into the clean nest part, it's actually attacked by the others. So, yeah. So, another extremely nice example, I think, what shows what I mean by social immunity is the so-called social fever that the honeybees are producing. And what they are doing is that a lot of individuals at the same time they can produce muscle heat, so they use their wing muscles but they don't fan their wings at the same time. And this means that because they're all doing that simultaneously, they can increase the hive temperature and produce some kind of fever at the colony level, at the superorganismal level. And this is then reducing the multiplication of particular pathogens in the colony, and thus they cannot multiply and not infect others. And when I was talking also about this interaction modulation, what termites are doing, for example, is that they quarantine their sick individuals when they are infected with nematodes, and they actually build walls around them. You can see here now a termite nest that is actually composed of the feces of the termites, which interestingly enough have antimicrobial properties. And so you can see a lot of walls already here, and what they are doing then is that they close these infected individuals and thereby exclude them from the active colony life. And so this was all very broad examples and not really my work either, but just to give you an example that actually all of these social insects are performing these types of collective defenses. And now I would like to give you a bit of an example of my own work on the ants, on epidemiology. In social networks, some very briefly on social immunization and on hygienic brood care. So first of all, a few methods. So what we are doing, of course, we first have to collect the ants to bring them to the colony, to the lab. And for this it's quite important because there are 14,000 different ant species that one picks the right species, so you have to have some knowledge of the populations and of the ants, and you have to have permits to collect the ants, and then you can use some kind of exhausters where you are sucking in and hold it towards the ants, and then you can collect them in glass vials. You can then bring them to the laboratory and rear them. We rear them simply in small plastic boxes like Tupperware with a plaster ground in which we can make some indentations because they like very confined spaces. And then we feed them with sugar, like honey or sugar water, and some cockroaches or other insect food, because the adults require only some carbohydrates, whereas the brood requires proteins. To be able to observe the ants and distinguish them, we color mark the individuals, or we can put some wires around their waist, and then we can observe them, as I've already mentioned, in quite a lot of replicates. We also now perform some video analysis. So we try to develop some tools for ant tracking, so that we can really get the trajectories of the individuals, and then you can also make heat maps and see in which areas the ants have spent most of their time, and when they have encountered and whom. And we also perform chemical analysis with gas chromatography, mass spectrometry, because ants are relatively blind. They are living underground, so their most important communication is chemical communication. And for this, we study their cuticular profiles, so the smell of the individual ants, which is actually quite different from different castes and different ages. And also we study the antimicrobials that they are producing. And lastly, we also use genetic methods like quantitative real-time PCR, which allows us to quantify, for example, pathogen loads, or look at the gene expression of the immune genes that are relevant in disease defense. And we also use some specific immune assays, some functional immune assays, where we, for example, have fungal growth in these microtiter plates, in these individual wells, and then we add some ant material, for example, some antimicrobials that they are producing, and then see how efficiently they can reduce this fungal growth. And with all these different methods, we have then studied the disease in colonies, and we are mostly interested in how sick and healthy individuals are interacting in a colony. And what we of course also do, we are not only interested in what happens when we have some disease in the colony, but you always have to compare it as closely as possible to some kind of control treatment. And for this, what we do is some kind, it's called a sham treatment, where here we, for example, apply to an ant some fungal spores in suspension, and then we use the same what we typically use here as a solution in which we are suspending the spores, simply this as the control treatment. And then we keep these treated individuals together with naive workers in small subcolonies, and we, for example, study then the antiseptic behaviors that we find. And one very important one is the grooming. For example, or very similar to the delousing that one knows from primates, also the ants are grooming each other. They are licking the body surface of their nestmates, and thereby they are reducing the risk of getting infected after an exposure to the pathogen. And we have recently also found that they don't only mechanically remove these fungal spores, but they in addition then also perform some chemical disinfection, and I will come back to that at the end of my talk. So we then also want to study how having lived together with a sick individual affects the individual immune system of those nestmates. And for this, what we have been doing is that after 5 days of the social contact, we then challenge the group members. So we basically then expose those nestmates that have previously been naive to this fungus. And then we measure their survival. And you can see here that this green line, these are the nestmates of fungus-exposed individuals as compared to the nestmates of control individuals, actually have a 1.5 times higher survival chances when they then get challenged with the pathogen. So that means that having lived together with a sick individual is somehow protecting those nestmates and reducing their susceptibility. So we were, of course, interested how this is actually achieved and which mechanism is contributing to this, what we have called social immunization in these groups. So to study this, we wanted to know what is actually transferred. Is there something that is transferred between from an exposed individual to their healthy nestmates? And in general, they could either give a signal or they could transfer the pathogen, and then the other individuals would raise an individual immune response similar to vaccination, or they could also transfer their immune effectors, so like the antibody parallels in insects. So what we did is that we used fluorescently labeled fungal spores, and we knew that our nestmates have been free of those before the experiment, and then we checked after 2 days of social contact, and it's now a bit difficult to see, but here you can see a leg of an ant and an antenna, and here the red dots are these fluorescently labeled spores. So we know that particularly through this grooming behavior, we have a transfer of the pathogen through the colony. But this transfer of the pathogen does not necessarily mean that those individuals really get sick, because this fungus is infecting insects by attaching to the cuticle and then penetrating actively through the cuticle. And before it's penetrating, it can actually be groomed off by themselves, but also by the nestmates. And but we found actually that there are internal infections of these nestmates that are mini infections that are very much smaller in intensity than what we find in the directly exposed individuals. And we then, and this is now just an example, we also then compared their immune gene expression and found that the nestmates of fungus exposed individuals have increased immune gene expression in these candidate genes that are working against fungal infections, whereas they do not have any increased immune gene expression of genes that are helping against bacterial infections. So that means that they have quite some specific protection against fungal diseases. And this is actually something that we don't only know from ant societies now, but also gives some parallels in human societies that take advantage of this active immune stimulation that one gets after low-level infections. And that is what has been used in early human medicine under the term of variolation or inoculation. For example, starting in the 9th century in China, people have taken infectious particles from the pustules of patients that had smallpox, put it into glass vials, then used it scratching the body surface, particularly of kids of rich people, and then rubbing some of the infectious material into it, or also in the nose, and thereby hoping to induce a low-level infection. Only that gave a lifelong protection, and this also worked quite well in 95% of the cases, but 5% of the kids developed a full infection and then died of it. So it is a risky strategy that works often, but it has then been replaced by modern vaccination, where one does not take live pathogens but killed pathogens or attenuated strains of the pathogen. And the ants, obviously, they could not yet sterilize their pathogens, so they are stuck with the variolation. So I've now talked about our work on interaction between sick and healthy adult colony members, and I would now also like to introduce what they are doing when their brood is infected, because what you normally see when you open up an ant colony is what people are calling the ant eggs. That's actually the pupae. So they have very small eggs, then they have larvae, and then at the last stage they have a pupa, often in a cocoon, but not in all species, because they're holometabolous insects. So this is what we have here, one pupa that we have exposed to the pathogen. And we then also studied their hygienic behaviors towards this brood. And again, what the nurse workers are doing is that they are grooming the brood, so they try to remove the infectious particles. And so we have then found that this is not only mechanical but also including some chemical disinfection. And I would now like to lead you a bit through the different steps how we found out about this. And so first of all, as I said, we typically have this fungus exposure treatment, and then you have to have control treatments that are as close as possible to the situation that you're interested in and mirror everything except so that the only difference is here the addition of the pathogen. So we had an absolute negative control with no treatment at all. We had this sham treatment where we applied the solution, in which we then here also have added the fungus. And we have realized so quite a lot of these ants that you find here in Europe, they belong to the formicine ants, and these formicine ants, they are for example the wood ants and the garden ants, and they are the ones that do not sting, but they are spraying formic acid when a predator comes into the nest. So that's their poison. And we had gotten the idea that this poison could also be used in the defense against pathogens, because it's actually used in meat preservation because it has antimicrobial properties. And the poison of the ants consists to 60% of this formic acid, and then it has also a few other compounds like acetic acid. So we have then used a pH, so here the gray thing here means that we placed the brood items on pH sensitive paper that is normally blue. It's a litmus paper, and it receives a red coloration when it comes in contact with acidic substances like the poison of the ants. And so we could extremely simply already show that there is a difference in the fungus exposure treatment, because here the litmus paper has turned very red in these 24 hours of our experiment, whereas there was only very little red in the two control treatments, meaning that the pathogen presence seems to induce the poison application. And we could also show that the poison application is very drastically reducing the germination ability of the fungus by like 50%, for example. So we were then interested in how are the ants actually applying that poison onto the brood. And so we made some behavioral observations, and we found that they are spraying the poison. So they basically, this is called the acetopore. The acetopore is the opening at the rear end where the poison can be, the poison droplet emerges, and then it's highly volatile, and it's then emerging, and it can also be directly sprayed. So we see that upon fungus exposure, the ants are applying this poison by spraying it directly onto the pupae. However, this is an extremely rare behavior, and it only occurs once every 4 hours in the pathogen treatment, and not even once per day in the controls. So we wondered whether this can really be enough to show these really huge differences that we have seen in the pH paper. And by studying more details, we also found a second indirect application. Namely, what the ants are doing is that they are first grooming themselves on the acetopore, so they take up the poison into their mouth, and they do this behavior of acetopore grooming more intense, but in this case it was not significant. In a second experiment it was significant, but at least you see that they are taking up the poison into their mouth, and then they are applying this poison by activated brood grooming towards the brood. And this is occurring very frequently. But first it was not so obvious to see it, but then we realized that they are actually often making this self-grooming, and because they are not extremely accurate, you can also see that on the pH paper, because there's some spillover. And I brought a video. Now I'm not sure whether you can very easily see the difference between blue and red in this video, so just in the beginning, like immediately when I start the video, you will see that the ant is bending its abdomen towards the mouth and is then spraying and does this acetopore grooming, and then later on it will continue the brood grooming. So here you have the head of the ant, and here you have 2 treated ant pupae. So here it's doing now this oral uptake, and you can also see that here around this area the pH paper is changing its color immediately, and then it is continuing to apply the poison and at the same time removing these particles from the brood. So we also showed directly that they are taking up the poison and have it in their mouth. And for this we also did a chemical quantitative analysis of individuals that have been treated differently. So we had a normal worker that was simply just taken out of the colony, but then we also had workers in which we glued the acetopore. So we just took normal glue and closed this opening. And then we either waited for 1 hour or for 24 hours. And by gluing this opening, we prevented that the ants can replenish what they have in their mouth. And so you can see here, when you quantify it, that by this closure and waiting for 1 or 24 hours, the poison, the formic acid content in the head is actually decreasing over time. So this only works if they really can replenish it very fast. And to show you what this means then for the inhibition capacity against fungus, you can see that this is directly correlated with the content of the poison in the mouth. So with acetopore blocked for 1 hour, it was not significantly different in both cases, but when we waited for 24 hours, then both the poison content in the head, in the mouth, and also the inhibition of fungal growth that they could prevent, that they could do towards the brood, was reduced. Then we of course wondered, what is the potential benefit of this poison uptake, because we wondered why they're doing that, because they're actually dying of formic acid themselves. If they are not if they are in a really contained small glass area and they panic, they spray a lot of formic acid and then they all die within a couple of minutes. So it's actually a bit of a dangerous thing to do, but we think that they have their mouth also covered with cuticle inside, so it seems to be okay for them to do it, and it has a lot of benefits because you can be much more accurate. When you spray, you're really losing a lot. You can better distribute it across items. If you can only spray it to a single pupa every 4 hours, you better take it up in your mouth, and then you can go from one pupa to the next fast. And there are also additional benefits to both the groomer and the colony. So we also saw that the ants often, when they directly after they have taken it up into their mouth, they clean themselves. So normally as a cleaner you have a quite high risk of contracting the pathogen, as we have shown before. This seems to be that they do not only use this chemical disinfection against brood items, but also against themselves. And also what happens to all the infectious particles that they are taking up. So they are removing it and they store it in their own mouth until they spit it out later in a compacted form as so-called infrabuccal pellets. So I show you a video and that you have to look very carefully. Here is the head of the ant, and first it's grooming the pupa, and then it spits out this infrabuccal pellet. And this is basically only compacted fungal spores. So first it's performing direct spraying, and it will then move a bit away from the pupa. and then really just take it out of its mouth in a second or so. And then, yeah, so now it's cleaning itself. And now it comes out, and they take it into their mouth parts, and then they place it away from the brood. And so here you can see this infrabuccal pellet that is expelled. So we have then looked at what happens with the pH paper around that pellet over time, and that's 20 seconds. And yeah, if the colors are good enough, you can see that there's actually some red around here. So that means that we then thought that they also use this formic acid that they have taken up into their mouths to clean those pellets. And what we have done to show that is that we we then basically collected these fungal spores, plated them out on an agar plate, and showed how many of these can still germinate. And we collected the pellets of ants that had been untreated, which are these, and others where we had depleted, which we had depleted from their poison. And then you could see that the germination inhibition was much higher in the normal workers. So that means that they do not only apply it to the others and they're inhibited on the brood, but they also clean their nest environment and themselves by this oral poison uptake. And yeah, with this, have talked for a full hour. I'm now at the end of my talk. I think I could show you that really in this collective disease defenses, behavior and physiology are really tightly interacting. And of course, this work is not possible for an own person, but this is my team and the funding sources. And I thank you for your attention, and I'm happy to take questions.